Hello everyone, please let me know if I'm audible. Okay, so we have reached our session 4 for your metabolism, and today we will be covering TCA cycle. Okay, sorry for that disturbance. I was just checking. Okay, yes. Hi Hina. Good evening. All right. So, before we begin, if you are not yet following me at an academy, you can do so using the given link. The link is also given in the description below. And if you have not yet signed up for the app, you can download the Unacademy Learning app from Play Store and you can use the code SPLive10 to start accessing the free resources. Okay, so there are a lot of free classes available on the platform which shall help you in your preparation of IIT JAM. There are also a lot of YouTube sessions that I have taken that might help you enhance your preparation. The watch list link shall be given in the description below. These are some of the plus courses that I have taken. Okay, so an Academy Plus is a paid platform wherein the only difference between the plus classes and the structured classes is that those plus courses are usually detailed and they are structured. So you do not need to go to other platforms or rely on any other material for your preparation, including the tests and quizzes. So, we take care that you have practiced enough before you go and write your exams. You have revised enough. So, we have provided unlimited access and also access to classes of math, physics, and chemistry, since that is also an essential part of your IIT JAM biotechnology. The classes will be live, same as the free classes that we have. They will provide you a platform to engage with us, ask doubts, and answer all the polls while the classes are going on. These are the subscription costs, guys. So one new thing that has come up is that along with the 6 months subscription you get an additional 1 month free, and along with the 12 month subscription you get an additional 2 months free. So if you are in second year or first year, you can go for either of these. And if you are in if you are going for IIT JAM 2022, then I think this is the best deal for you. You get 7 month subscription at a low cost. So when you watch these free classes, you will get some credits, okay, which you can use to avail discount on this particular amount. Additional to that, you can get a flat 10% off when you use the code SPLive10. If you feel that any of the content that we are teaching on YouTube is inappropriate or it is incorrect, you can report it and you can claim your prize. What you have to do is you just have to click on the description link that is present uh, in the description of this video. Okay, so I already told this to you that you have. Okay, this offer is valid only till 29th of July. After that, you will not be able to have access to this. So go and subscribe if you want to. Okay, coming back to the topic of discussion. So we are currently studying your biochemistry unit. The major chunk of it is metabolism part. So metabolism includes quite a lot of topics like your glycolysis, TCA cycle, their regulation, Then you have photosynthesis, you have nitrogen fixation, urea cycle. Yes, I think that pretty much covers about 90% of your metabolism part in your IIT JAM 2022 syllabus. So, guys, we have finished with photosynthesis in the YouTube sessions and in the special classes as well. So, you can go and access either one of those. Today at 5:30 p.m., we will have another session wherein I will tell you the differences between C3, C4, and CAM cycles. ठीक है. That's really really important. Plant physiology में से बहुत सारे exams हैं, not just IIT JAM wherein you get questions from C3, C4 and CAM. There are other exams as well. So do attend this session to know what is the difference and what are the parts that you need to focus on for your exam. All right. Uh, so this session is at 5:30. There's another session at 7:30 which is a practice session. All right. So for those of you who are preparing for GAT B, there's a practice session. Please make sure that you attend, because what happens is you have a live poll there, and we have a leaderboard system so that you will know that where do you stand in class, what is your rank, you can compare your performances. Okay. All right, coming to TCA cycle, citric acid cycle or your TCA cycle, can somebody think of more names? I have already told you two names. Does this cycle have any other names? Yes, no, no answers. Come on, be quick. It's a very easy question. Yes, Isha, that's right. Krebs cycle. Okay, थोड़ा सा lag होता है when you write the comments, so please be a little patient while I take your names and while I get to the answers. So yes, you're right. It's also called as your Krebs cycle. Why is it called as Krebs cycle? Yes, very good. It is named after the scientist who discovered the cycle, that is Hans Krebs. Uh, so the citric acid cycle here refers to the first molecule that is formed during the cycle's reaction, that is citrate. So the first product that we get here in this cycle, that is citrate or your citric acid. Okay. That is why the name citric acid cycle. Tricarboxylic cycle is because of the three carboxyl group on its first two intermediates. That's why the name TCA cycle. And Krebs cycle, after its discoverer Hans Krebs. This here is the second step of your respiration. It occurs in the mitochondrial matrix. It is catalyzed by a series of enzymes that are located in the mitochondrial matrix, which is obvious. Exception is succinate dehydrogenase. Now, please remember, guys, there are a lot of questions that can be framed when it comes to your exceptions. Okay, so please make sure that you are very very thorough with whatever exceptions I am teaching you, or whatever exceptions you come across. Maybe you can make a chart or something and stick it in your room, or you can have it on the first page or the last page of your notebook so that you remember it. Okay, so the main thing is remember whatever works for you. Sticky notes, charts, jo bhi jaise bhi yaad karna ho. But please remember these exceptions. Okay, like this question has been seen in so many exams so many times, right? That all of the enzymes that are involved in your citric acid cycle, they are located in the mitochondrial matrix, and the only exception to this is succinate dehydrogenase. Okay, next thing is pyruvate generated in the cytosol during glycolysis is transported into the mitochondrial matrix. So we will see how your pyruvate is being generated in the glycolysis. Once we study glycolysis, we are yet to cover glycolysis. I started with TCA cycle because this is easier, and I think somebody of you had requested for the video. So this is transported into your mitochondrial matrix. Pyruvate is decarboxylated. That means removal of carbon dioxide is happening to your acetyl coenzyme. So this pyruvate that was being generated in the glycolysis, here it is getting decarboxylated. Carbon dioxide is being removed, and that is passed on to your acetyl coenzyme A. Acetyl coenzyme A is thus the connecting link between glycolysis and the next series of reactions that yield more energy in the form of ATP. So this part here is very very important. Okay, a question can come from here, that what do you think is the link between glycolysis and the subsequent steps of respiration, right? So your answer should be glycolysis. Sorry, acetyl coenzyme A. ठीक है? Is it clear till here? Please give me a yes or no, or if you have any doubts, any questions. Okay, great. So then moving ahead to your mitochondria. So like when we studied photosynthesis, we went through the structure of chloroplast. Here we need to go through the structure of mitochondria, because this particular step here is happening inside the mitochondrial matrix, right? So your mitochondrial matrix has a smooth outer membrane. It has an invaginated inner membrane. So invaginated means that it is thrown into folds which are called as cristae. Okay, so these folds are your cristae. These are the folds of the inner membrane. Matrix is an aqueous phase within the inner membrane. So can you see this pink portion here? This is your matrix. Okay, and there is an intermembrane space. So between your outer membrane and your inner membrane there is space because of the folding pattern, right? So because of the folding pattern, whatever space is left, that is known as your intermembrane space. So coming back to the citric acid cycle, how is it happening and what is it? So here you have pyruvate, okay, which is a product of what? Where are we getting this pyruvate from? Where are we getting this pyruvate from? जल्दी बताओ. I just told you. Right. First step of respiration क्या था? Glycolysis. Yes, correct. So that's your first step of respiration, from where you are getting your pyruvate. ठीक है, that's correct, Isha. Good. So once you get your pyruvate, first step जो होता है वो होता है decarboxylation, right? तो ये जो आपका acetyl coenzyme है A है, that will come here. Your NAD plus will get reduced and your decarboxylation is happening. Okay, so you have a removal of CO2, you have your reduction happening, and you have an addition of acetyl coenzyme. So basically यहाँ पे acetyl coenzyme A has been introduced, right? So here now what happens is it is undergoing another reaction in the presence of citrate synthase. So I asked you to figure out the difference between synthase and synthetase. In fact, I taught this to you in the enzyme class. So who knows and who will answer? Let me know. Meanwhile, I'm moving ahead. So here you have your acetyl coenzyme A, which will undergo the action of citrate synthase, thereby releasing the coenzyme A part and converting into the first stable product that we have, which is citrate, right? So that is why the cycle, citric acid cycle, right? That's why the name. So now that we have citrate, it goes into formation of another intermediate compound, which is your which is your isocitrate. Okay, and this is happening in the presence of an enzyme known as aconitase. So this here is your isocitrate. So citrate and isocitrate, they are nothing but isomers of each other. Okay, Isha, you have, yes. Yes, you are correct. Synthetase requires ATP, and your synthetase, it does not require ATP. ठीक है. So yes, your isocitrate then will undergo a dehydrogenase process, wherein it will be uh, and a decarboxylation, right? So यहाँ पे again your NAD plus is getting converted into NADH. Your isocitrate is getting converted into 2-oxoglutarate. No, sorry, this is the enzyme. This two here is 2-oxoglutarate. Okay, so why am I getting? Okay, I think the diagram is a little mushy here. So I will just do what? I'll just highlight first the products that we are getting, so that you do not get confused like me. Yes, now we are good to go. So what happened was you have your pyruvate, which is undergoing reduction and decarboxylation to form a acetyl coenzyme, to have an acetyl coenzyme A molecule here, which from which your coenzyme A will get removed, get converted into citrate. Citrate gets converted into isocitrate. Isocitrate gets converted into 2-oxoglutarate, and then your 2-oxoglutarate gets converted into succinyl coenzyme A by the action of enzyme 2-oxoglutarate dehydrogenase. Now again, this is removing. Dehydrogenase means, yes, alpha ketoglutarate is another name for it. So if you like to remember that, you can remember that also. I don't mind. ओके, सो डिहाइड्रोजनेस मतलब क्या, दो हाइड्रोजन एटम्स जो हैं वो रिमूव हो रहे हैं, ठीक है, from your 2-oxoglutarate, so then it is getting converted into succinyl coenzyme A, okay, which further is exposed to the action of succinyl coenzyme A synthetase. Now please remember, this is not synthase, this is synthetase, so there'll be a requirement of your ATP here. As soon as this is using up your ATP, what is happening is it's forming a succinate molecule, which again undergoes a dehydrogenase reaction and it's getting converted into fumarate. Fumarate is getting converted into malate by the action of fumarase. Malate goes into forming oxaloacetate, which is again because of my malate dehydrogenase, and oxaloacetate is again getting converted here, like oxaloacetate along with pyruvate, they react and they form the first product, that is your citrate. So that's your citric acid cycle or your TCA cycle or your Krebs cycle. You need to remember all of these. Okay, you need to remember the names of the enzymes, the kind of reactions that are happening, and the products that are formed. Okay, so what all do you need to remember? Products, enzymes, kind of reactions. At least this much you need. Okay, so basically in short, ये पूरा chart जो है ये important है. You cannot skip out on things. You cannot think that you'll study this, you won't study that. That's not happening. Malic enzyme decarboxylates malate to pyruvate and enables plant mitochondria to oxidize malate. So this is just information here about this particular enzyme. And I think that's all. One molecule of ATP is synthesized by a substrate level phosphorylation during the reaction by succinyl coenzyme A synthetase. Okay, so you can see here that ATP is being synthesized. ADP और phosphate जो है वो combine होके ATP बनाएगा and the energy is being provided by substrate level phosphorylation. So again, let's go through the steps in detail. Your pyruvate is decarboxylated in an oxidation reaction by the enzyme pyruvate dehydrogenase. So here what all is formed? Your NADH is formed, carbon dioxide is released because again that's a process of your decarboxylation, and acetyl coenzyme A is formed. Next step. So you have citrate synthase, that's an enzyme which combines with acetyl group of acetyl coenzyme A with oxaloacetate. So the oxaloacetate that we were getting is combining here with your uh, acetyl coenzyme A to give a citrate, which is a 6 carbon tricarboxylic acid group. Okay, citrate is then isomerized by to isocitrate by the enzyme aconitase. So this will form under an isomerization enzyme. The next two reactions are successive oxidative decarboxylations, each of which produces one NADH and releases one molecule of carbon dioxide, yielding a 4 carbon molecule, which is your succinyl coenzyme A. So isocitrate here quickly converts into 2-oxoglutarate, or as Isha mentioned, alpha ketoglutarate, and then it is getting converted into succinyl coenzyme A. At this point, three molecules of carbon dioxide have been produced for each pyruvate that entered the mitochondria. Succinyl coenzyme A is converted to succinate. The synthesis of ATP via phosphorylation reaction is catalyzed by succinyl coenzyme A synthetase. Succinate is oxidized to form fumarate by succinate dehydrogenase, which is the only membrane associated enzyme of the citric acid cycle, and also the part of electron transport chain. So I already told you that this falls under exception. All the other enzymes that we are using, all the other enzymes that we are using are what? Uh, they are this thing. They are of your mitochondrial matrix, right? If there is a question that which enzyme from your TCA cycle is not a part of the mitochondrial uh, matrix, then you'll have four options. You have to choose succinate dehydrogenase because it's a membrane associated enzyme. It is not uh, present in the this thing. It's not present in your matrix. Okay. Okay, Isha has posted a question. Malonate is an analog to some intermediate. Which is it? Uh, Isha, what do you mean by analog? I mean, see, like, in terms of what? In terms of acting as a substrate for the enzyme or what? क्योंकि malonate का जो structure होता है वो कुछ ऐसा होता है. I'll show you. It's something like this. You have, so these are your carbon atoms, okay? So you have three carbon atoms, and naturally this will be attached to your CH and CH. So obviously it is similar to your, please remember I use the word similar, I did not use the word same. Okay, there's a huge difference between similar and same. So here you have malonate ka structure which is similar to your malate. So maybe if you are saying that it is a analog to some, it could be closest to malate, not anything else. ठीक है. So the citric acid of plant has some unique features. But again, Isha, I am not sure, and I can check it up and get back to you. But malonate, as a part of TCA cycle, I have not come across it. But like you're saying that it's an analog, so I'll just read more about it and let you know in class. Okay? I hope that works for you. So the step catalyzed by your succinyl coenzyme A synthetase produces ATP in plants and GTP in animals. So that is one of the difference. Okay? A feature of the plant citric acid cycle that is absent in many other organisms is the significant activity of NAD plus malic enzyme, which has been found in the matrix of all plant mitochondria analyzed to date. Okay, so this is something that is common in all the plants that we know of so far. All the hydrogen atoms from the original glucose are now on the hydrogen carriers, that means NAD and FAD. These hydrogen carriers enter the next phase, which is known as the respiratory chain or the electron transport chain for further release. So what all we did so far, I mean the steps of respiration, glycolysis, then we came to TCA cycle, and now we are moving to electron transport chain. सो इलेक्ट्रॉन ट्रांसपोर्ट चेन मैंने डिस्कस करी है ऑलरेडी स्पेशल क्लास में. आई जस्ट गिव यू अ क्विक synopsis again. The electron transport chain catalyzes an electron flow from the NADH and your FADH2, which are again your hydrogen carriers, to oxygen, which is the final acceptor of the respiratory processes. Your ETS in plants contain multiple NADH dehydrogenases and an alternate oxidase. It catalyzes an electron flow from NADH to oxygen, which is again the final acceptor. The process is carried by four membrane bound enzyme complexes in the inner mitochondrial membrane. So this is really really important. Okay. So there are four complexes. In this session, we are not going to go into details of it because this is again a very very long topic. But from this diagram you can see that how your electron transport chain is working, right? You have four different complexes which are performing different functions. They have pumping of hydrogen in and out of the system, and ultimately what is happening here is your oxygen is accepting the electrons. Okay. So all of this we will do, but first let's check if you have understood what we did so far. So there's a question on your screen. Please answer it. So the question says the end product of glycolysis is pyruvate, which enters the citric acid cycle after being converted to what? Yes, very good, Isha. It is acetyl coenzyme A. Next question. So these are very simple questions just to check if you guys were paying attention. The first intermediate in TCA cycle is what? The first intermediate. Yes, correct. The answer is citrate. Which of the following enzyme does not take part in the TCA cycle? Yes, very good. The answer again is C, which is pyruvate dehydrogenase. Now, please remember that before we enter the TCA cycle, what happens is pyruvate gets converted to acetyl coenzyme A, right? And it is acetyl coenzyme A that is entering the TCA cycle. And this particular enzyme is working here. Okay. Whereas all of these other three enzymes, they are inside your TCA cycle. They are happening within your TCA cycle. So the answer here is pyruvate dehydrogenase. Okay, which of the following is not the intermediate of Krebs cycle? Yes, it's the linking step. Very good, Isha. Yes, very good. It is C, it is isofumarate. So isocitrate, succinate, and fumarate, they are the intermediate steps of Krebs cycle or your TCA cycle, but this one is not. Okay, this we've not done. Yes. In Krebs cycle, hydrogen of malate is accepted by which one of these acceptors? FMN, FAD, NAD, or coenzyme A? If even if you're not sure, you can at least like just try. Okay, Isha, you are saying B. All right, B is FAD. Okay, no, no, that's not FAD. FAD is actually an acceptor when your succinate is turning into fumarate. Okay, here the answer is NAD. Okay, next question. Most enzymes that take part in your Krebs cycle are located where? Okay, I see some of you joining in. Please answer. Most enzymes that take part in Krebs cycle are located where? Yes, the answer is B, which is mitochondrial matrix, except your succinate dehydrogenase. Okay, I think I told you the answer, but okay, go ahead. Enzymes of TCA cycle are located in the mitochondrial matrix, except one which is located in the inner mitochondrial space. Name the enzyme. Yes, very good. Isha, it is succinate dehydrogenase. Okay, so I think we are done with with this topic. The questions might seem easy to you right now because you freshly done the topic, but what happens is after a point of time you tend to forget. So I would suggest that please practice this cycle by writing. Okay, Don't just study it or don't just read things, but only when you write it and try to write it without looking at the diagram, only then you will gain the confidence that okay, now you can finally know the TCA cycle. But most physiological cycles are not this tough. However, if you write it down a couple of times, I'm sure you'll be able to memorize it soon. Okay, so uh, other than that, if you are signing up for the Unacademy Learning app and you're new, so you can use my code SPLive10 for signing up and using the free resources, also getting a 10% discount on your plus subscriptions. Today we have a session at 5:30 p.m. For C4, C3 and CAM, and also we have a practice session at 7:30 p.m. for GAT-B students. So I hope to see you guys there. That's all about the session for today. If you feel that the video was helpful for you, please hit the like button, do share